Good morning, students. So today's session we have to talk about one of this very interesting as well as very important topic, that is blood groups. Okay, and especially we are going to talk about this human blood grouping system. Okay, so there is two blood grouping system will be followed throughout the world to categorize the blood group of human beings. Okay, so human beings blood group is not similar to everyone, that we knows, right? And it is dissimilar for everyone, and basically there is four types of blood group is said, right? And let us discuss about these four blood groups and how it will be categorized or classified, right? And that is the first thing about this. And the blood grouping system is ABO blood grouping system. Okay, and the second method is RH factor blood grouping system, right? So this is the two blood grouping system, RH factor system as well as ABO system we are following now throughout this world, right? And let us discuss about the first system. It is ABO blood grouping system. Fine. So anyhow, before we move to this topic, and you must know about the two terms which is the key for this blood grouping system or the basic for this blood grouping system. That is antigen as well as antibody. Antigen as well as antibody. So hope you are hearing these terms newly. And antigen, if we say in a simple manner, antigen is a substance which is coming from outside and entering into the body. So whatever is not our own, whatever is not own to the body is considered as antigen. Or we could say it is the foreign particles. Foreign particles. So whatever it may be. It may be as a pathogens, or it may be as a drugs, or it may be as a food. Whatever it is which is entering into the body is known as antigen, right? So when one antigen enter into the body will produce automatically one substance for matching of this antigen. That is known as antibody. So when one antigen enter into the body will respond for this antigen. So the body will synthesize one substance, that also one protein substance is known as antibody. So this is a proteinaceous substance. So this will produce this, it is a protein substance. This protein substance will be matched for this antigen. Have you got? For example, if it is antigen is X, just for example, if the antigen is X, the antibody also will be X. Antibody also will be X. So this X and the X will react together. It will be like as, like lock and key, we could say. For example, if there is any lock, we can open the particular lock with the particular key only. We cannot use any other keys to open the particular lock, right? So it will match each other. So likewise, for this particular antigen, the particular antibody will be produced. This only will react together. So this is the basic for, don't forget this one. So this is the basic for this blood grouping concepts. Understand? So the antigen and antibody reaction, right? So the particular antigen will react with this particular antibody, the matching antibodies. Have we got? Okay. So now we can come to the point about this ABO blood grouping system. So ABO blood grouping system. Fine. ABO blood grouping system. First system, blood grouping system. Okay. So this system will be followed as per the presence or absence of this one of this antigen which is present on the surface of these RBC cells. Do you get me? For example, listen. So presence or absence of the particular antigen which is present on the RBC cell. So if it is a RBC cell, consider if it is RBC cell, RBC. So this surface of this RBC cell, where is the RBC cell? RBC cell is present in the blood. There is 3 types of cells we said in the previous class, fine. And it is present in the plasma, right? It is present in the plasma, right? So this is the RBC cell. So the surface of this RBC cell, consists of the antigen. Okay. So this antigen is 2 types. This antigen is 2 types. That is A antigen as well as B antigen. Are you following me? A antigen as well as B antigen. Okay. So as per the presence or absence of these 2 antigen, the blood grouping will be made or classified for human beings as per the presence or absence of this antigen. Do you get me? So this antigen is present on the surface of all the RBC cells in the human blood. Okay, so there is 2 antigen is A, there is A as well as B. So now we can classify the blood group as per the presence or absence of this antigen. Do you get me? So first we can consider it is, A, blood group A. So here we could write as blood group. Blood group. Okay. So here the blood group is A. Blood group is A. So which means, do you understand what will be the antigen here? The antigen is, here the antigen will be answers A. Do you understand? Here the antigen is, if the antigen is A, the blood group will be considered as A blood group. As per this name of this antigen, the blood group will be considered. So the antigen is A, so the blood group is A. Have we got? Okay. Then, if it is B, so if the antigen is B here, the blood group will be B. So what will be the antigen here? Antigen will be B. So the blood group also B. Okay. And one more. There is AB blood group. Could you say what antigen will be here? Yeah, of course. Both the antigens will be there on this surface. Both the antigen will be there. So here antigen we can write it is A as well as B. Okay. And what is the one more blood group? Could you tell me? That is O blood group. Right? So there is O blood group. So O blood group in the sense there is no antigen. So in this surface, there is no antigen will be, in this surface there is no antigen will be present. So it will be considered as O blood group. So the antigen is none. No antigen. Okay. Then we have to talk about the another one very important thing. So we said about the antigen now. So one more term already we said it is antibody, right? So where is this antibody? So this antibody is present in the plasma. Natural antibodies. Otherwise we say when the antigen enter into the body, the antibody will be produced, right? But here naturally the antigen, naturally present antibody is present in the plasma. Naturally present antibody is present in the plasma, right? So as I said already, if it is a matching antibodies and if it is matching antigen and the antibody, it will react. So the antibody must be the opposite one. Do you understand? Naturally it is present. So if it is the same antigen and the antibody, it will react each other. Okay. So it is opposite. Antibody will be opposite one. Have you got? So now we can write this antibody here. Antibody. Okay, antibody. Then could you tell me now what are this antibody may be present for this antigens? So listen, if it is A antigen here, the antibody will be mentioned as B antibody. Anti-B. Anti-B. Do you understand? Okay. And could you tell me if it is antigen B, antibody will be A, anti. A. Have we got? Okay. Then if there is both antigens, if it is both antigen is present on the surface, then what happens there? Listen. Here A antigen is there as well as B antigen also there, right? So for example, here A antigen, this is B antigen. In case if there is this, A, B antibody here, this both will react. If it is A, this A may react here. If it is B, B will react here. If it is both will react there. That's why there is no antibody here. Do you understand? There is no antibody here. Do you get me? And so O blood group, and the now last one is this O blood group. O blood group, there is no antigen, we said, right? No antigen is there. So these cells, these RBC cells, since there is no antigen, when in one antigen enter into the body, and it will be protected by both the antibodies. Understand? The antibody will protect if there is any antigen enter into the body. This body will be, or the cells will be protected by the antibodies. Antibody only is the protecting substance. It says it will come under this immune system, antibody, right? So this O blood group consists of both antibodies. So if A antigens comes, this A antibody will react. If B antigens comes, B antibody will react. Do you get me? So this is this method how this blood grouping is categorized as per the presence or absence of this antigen A and B. So this system is known as ABO blood grouping system. ABO blood grouping system. Okay. Now we can talk about another one matter that is regarding this, blood transformation, right? That is also one of this very important area. But blood transformation, donation, or we could say as a blood donation or transformation. Donation. Donated to whom they can donate it, right? So listen. So this is the point you have to keep in mind always. A antigen antibody react, but it should be as a matching one. Have we got? So it should be as a matching one. You keep it in mind. So now we can come to this point here. To whom they can donate this blood. So the same blood group they can donate, that is doesn't matter, right? Because the antigen and the antibody will be same. So no problem will be there to donate for the same blood group persons, right? So the same blood group we can write for everything. So blood group A persons can donate to A, right? And to whom they can donate. So listen, just you consider about this antigen. Here what is this antigen? A. So it will react with this A antibody, right? So when this A blood group is donated to the B, here the antibody is A. So it cannot donate because this A antigen and the A antibody will react together. So it cannot donate to the B blood group, right? Then if we donate this A blood group to the O blood group persons, here also A antibody is there. So this A antigen and the A antibody will react together. So we cannot donate this blood group to O person also, right? But we can give to this, just to watch this AB, there is no antibody at all, right? So when this antigen enter into the body, there is no antibody to react. So we can donate this one to AB group. Have we got? So we can donate this one to AB group. Understand? And the next one is B blood group. So here also you watch, this antigen is B, right? So we can check to every blood group. When we give this B to A blood group person, here antigen is B, meanwhile here antibody is B, so we cannot give, right? And same you come to the O blood group, here antigen is B, here antibody B also there, right? So we cannot give to O blood group persons. But of course we can give to AB blood group persons because there is no antibody. So to whomever they can donate, B blood group person, whomever they can donate, there is B blood group as well as AB blood group. Have we got? Okay. And the next one is AB blood group. AB blood group, listen, here this antigen is AB antigen is AB. So if you donate to A group, here A group antibody is B. So here B is there. So we cannot give. Right. If we give to B blood group person, here antibody is A. So here also A antigen, here A antibody. So we cannot give. So because it both will react and the cell will be destroyed. Right. Then here O blood group person, here antigen is AB, here antibody is AB. So we cannot give to. So we can donate only to AB. Have we got? Okay. And the next one, O blood group persons. So O blood group, antigen is none. There is no antigen at all. So if there is antigen only, it can react with the antibody. So we can give this one to, so here we can, A also we can give because there is no antigen here to react with this antibody. We can give to B because there is no antigen to react with this antibody A. And here there is no antibody at all. Understand? And to O also we can donate. So to whomever it can donate, O blood group, A blood group, B blood group, as well as AB blood group. Okay. So have you noticed one thing here? AB is a universal recipient. Universal recipient, right? So because everyone can donate to AB group. So AB can receive from everyone. That's why AB is considered as universal recipient. It can receive from everyone, right? It can receive from A, it can receive from B, it can receive from O, and it can receive from AB also. So it is considered as universal recipient. But just to notice this O blood group can give to everyone. It can donate to everyone. So listen, it can donate to O group, it can donate to A group, it can donate to B group, and it can donate to AB group also. So, and O blood group will be considered as a universal donor. It can give to every papers. Universal donor. Have we got? So that's all about this ABO blood grouping system. And here one more term they added. There is antigen here, and here this antigen is considered as agglutinin. Sorry, agglutinogens, and antibody is considered as agglutinin. So it's agglutinogen. This antigen is considered as antigen is agglutinin. Sorry, agglutinogen, and antibody agglutinin. Agglutinin. Why? Because this name is given. So as I said already here, antigen and antibody will react. When it reacts, the cells, it will destroy, it will blast the cells. So that's as per the reaction, the clumping reaction, the name is given. For antigen, it is it is a clotting, we can say. A blood clot may form. When it reacts each other, the blood clot will form. And that only it is called as agglutinin or agglutination, right? Agglutination, that process. So this antigen is agglutinogen, antibody is agglutinin. So as per that, this name has been given, right? Fine. So let us go for the next blood grouping system. It is RH factor, right? RH factor. So this also based on this presence of another one antigen, that is known as D antigen. That is known as D antigen. Okay. So in ABO blood group system, we say this antigen is A and B. As per the presence of these two antigen, we have classified as 4 groups. Is it clear? But RH factor also one of this category we have to consider when we go for blood transformation. Okay. So either it is present or absent, it doesn't matter for this own body. But when we transfer the blood, we have to consider this RH factor also. Have we got? Okay, so let us see how they have classified this blood as per this RH factor. So this classification is made as per this one of this antigen, that is known as D antigen. Okay, so this antigen also present on the surface of this RBC cell. So on the surface of this RBC cell. So this antigen we can call as now. So this antigen now we can call as, D antigen. D antigen. Okay. So first this D antigen is they found in this one of this monkey, that is rhesus monkey. So first they have found it in the rhesus monkey. Rhesus monkey. Hence the name is given as RH factor. Do you get me? So why the name is given? Because this factor or this D antigen, first they found on the surface of this RBC cells in the rhesus monkey blood. Hence this name is given as RH factor. Okay. So now as per the presence or absence of this D antigen, the blood will be classified as two types. That is, one is RH positive, RH positive. The second one is RH negative. RH positive as well as RH negative. Hope you understood. If there is a presence of this D antigen, it is known as RH positive, and if there is no D antigen, it will be considered as RH negative blood group. Okay. So all around the world they said 80% of the people may consist of this D antigen on the surface of this RBC cells. So this is RBC cell, right? RBC. And rest around 20% of the people, there is no RBC cell. So they will be as RH negative persons, right? So it doesn't matter, as I said, but when they transfer the blood, it will be the one of this factor we must to concentrate, right? Okay. So in certain cases, one of the thing it is during blood transformation we have to consider this one, and another one factor it may collapse the disease when the mother is RH negative. When the mother is RH negative and when she is as a carrier, when the mother is as a carrier, which means during the pregnancy period, the fetus will grow inside this mother's womb, right? So if it is in opposite blood group, for example, mother is RH negative, but the fetus which is growing inside this mother's uterus as womb is RH positive. In such a case, maybe this RH positive factor the child may get from the father, right? So but the mother is RH negative. What do you mean here RH negative? The antigen, there is no D antigen. In the RBC cell, there is no D antigen. But in RH positive means in the RBC cells, D antigen will be present. The D antigen will be present in the surface. Am I right? So D antigen will be present. Okay. So in such situations, so if the fetus is carrying this, if the fetus is RH negative, it doesn't matter. But in case mother is negative but the fetus is positive, what happens here? In the first child, I mean RH positive child, there is no problems will occur in the blood, because mother's blood as well as the fetus blood will be separated by the placenta. It's a connective tissue. It's a connective tissue between the mother and the child. So the mother's blood and this fetus blood will not mix with this mother's blood. Have you got? So, but at the time of this delivery, because of this delivery, when they cut this placenta, what happens here? Maybe there is a probability to enter this fetus child, the fetus blood to this mother's blood. At the time of this delivery, when they cut this placenta, there is a chance to enter this fetus blood to mother's blood. Have you got me? So what happens here? When it enters, what happens here? And the fetus blood, actually what is there? D antigen is there. Am I right? When this D antigen entered into the mother, there is no antigen now. Right? So when this antigen, follow me. When this D antigen enter into the mother blood, automatically what happens? The mother body will produce D antibody. D antibody, because this is the antigen. As I said in the beginning, when antigen enter into the body will respond for this antigen and it will produce a matching antibody for this antigen, like a lock and key I said. Okay, so this is produced for this mother now. Now it is present in the mother, so no problem for the mother now, because it is present, that's all. Antibody is present. Okay, but in case of this second delivery, second child, so if she conceives the second child, what happens there? Now in the mother's body, what is there? Now in the mother's body, D antibody is there. D antibody is there. Now the fetus is RH positive. Which means, what do you mean RH positive? D antigen will be there. Am I right? D antigen will be there. Okay. So during the second pregnancy, surely already this mother's body is produced antibody because of this first delivery. So when she conceives the second child, this antibody will transfer from mother to the fetus. Okay. So already what is there here? D antigen is there. So this D antibody and the D antigen will react each other and it will destroy this RBC cells. It will react, so it will clump the blood, clotting of the blood. Do you understand what is clotting? The blood cells will clump each other, like you know when the milk is becoming as a curd, it clots, right? So likewise this blood will clot because of this D antibody as well as this D antigen. This both will react together, so it will produce the blood clotting. Blood clotting, right? So this only known as agglutination. Okay, so blood clotting will be produced. So because of this, what happened now? So the RBC cells will be destroyed. Why RBC cell is destroyed? Because this antigen is present on the surface of this RBC cell, right? So this RBC cells will be destroyed. So RBC cells destroyed. So this is leading to this anemia as well as jaundice. Okay, so and even if this condition may lead to the fatal to the child, it will be very dangerous to the child and the child may die. Understand? So this disease is known as erythroblastosis fetalis. Erythroblastosis fetalis. 1 minute. Erythroblastosis fetalis. The erythro means RBC cells. Blastosis means RBC cells will blast. It will destroy. Since it's happened in the fetus, it is mentioned as fetalis. So this the term, don't think it is a tough one. Erythro means RBC cells. Remember it like this. Erythro is RBC cells. Blast means it will blast, destroy. And fetus. So fetalis. Erythroblastosis fetalis is one of this very important question. Don't forget. Okay. So in this condition, and at which condition it happens? When the mother is RH negative and the fetus is RH positive, this condition occurs. Have you got? And how to avoid this one? So there is a treatment is there to avoid this situation. So what happens here? Immediately after this delivery, so when the child is removed or when the placenta is cut off, when the child is removed from the mother's womb, immediately they will admit this one anti-D antibodies, which means don't confuse already antibody is there in the mother's blood, right? Already D antibody only there in the mother's blood, right? So this antibody, when how it is produced? When the antigen is entering from the fetus to the mother. When the antigen is entering from the fetus to the mother body immediately started to produce this antibody, D antibody. Okay. So when they admit or when they inject these anti-D antibodies immediately after the delivery, means what happens here? Anti-D antibodies. This anti means this is against this D antibodies. Anti, have you heard this? We used to say this anti-inflammatory agent, which means it is against for inflammation. Have we got anti-inflammatory agent? Means it is against for inflammation. Likewise here, D antibodies. It is against for D antibodies. So already D antibodies started to synthesize in the mother's body. So when we admit this anti-D antibodies, this will destroy this D antibodies. Antibodies which is present in the mother. So this will destroy the D antibodies now. So if there is no antibody, there is no problem at all again. Have we got? So she can conceive the second child. So even the blood is transferred to this child also, it will not destroy these RBC cells of this fetus because there is no antibody. Have we got? So in such a manner, this second fetus will be protected. Have we got? And this D antibody is otherwise known as RHOCUM, RHOCUM. It is known as RHOCUM. Have we got? Okay. And we have to talk about one more point, that is the agglutinogen. Do you remember this term? Agglutinogen means the antigens, right? So antigens is known as agglutinogen, and this is made up of, this antigen is made up of the substances, that is sucrose, D-galactose, N-acetyl glucosamine, as well as 11 terminal amino acids. So these are all the things is present in the antigen. This antigen is made up of all those substances, right? And this attachment of this 11 terminal amino acid is catalyzed by this enzyme, that is glycosyl transferase. Glycosyl transferase enzyme is catalyzing this reaction, which means attachment of this amino acid on the surface. Have we got? So hope we can finish here. And today's topic is one of this very important thing, ABO blood grouping system as well as this RH factor. So RH factor, erythroblastosis fetalis, is one of the very important area, and how could we avoid this one, and what is the reason for this one also will be asked, right? And how this ABO blood grouping will be categorized. So that also one of this very important question. Fine. Okay, so please go through this topic, and for half-yearly examination till respiration topic is given. So if you leave this topic, you cannot pick up it later. So please you read out this topic and understand the concept first of all, and later we can learn. Have you got? So please go through the topic and get the content now, or concept first of all. Have you got? So hope you will do it, and we can meet in the next class with the next topic. Until then, bye. Thank you.